Welcome to Food Safety 5, monthly reports from the Food Safety Magazine editorial desk to keep you abreast of top news stories and current events impacting the food safety sector. This episode covers 4 new studies with implications for food safety and authenticity, including evidence of widespread avocado oil adulteration, findings about how microplastics and plastic food contact material characteristics may affect bacterial biofilms, and how stress adaptation traits could help E. coli persist throughout the farm-to-fork continuum. First, a new University of California, Davis study suggests that avocado oil used as an ingredient in many processed foods may not actually be pure avocado oil, raising concerns about ingredient authenticity and supply chain oversight. The study analyzed oils extracted from 74 commercially available chips, mayonnaise products, and salad dressings labeled as containing either avocado oil or olive oil as the sole declared edible oil. Researchers found that 89% of avocado oil labeled products exhibited compositional profiles inconsistent with authentic avocado oil. By comparison, only 1 of 20 products labeled as containing olive oil raised similar authenticity issues. Among the avocado oil labeled products, 93% of chips, 71% of mayonnaise products, and all salad dressings contained fatty acid and sterol profiles indicating the presence of other oils. To determine whether food processing could explain these differences, the researchers conducted laboratory frying and emulsification experiments. Those experiments produced only minor changes in the markers used to evaluate authenticity, suggesting typical processing conditions did not explain the discrepancies. Lead author and UC Davis professor Dr. Selena Wang said the findings point to the need for greater verification of premium oil ingredients throughout the supply chain. According to Dr. Wang, manufacturers may purchase edible oils through third-party brokers or multiple suppliers, making ingredient authentication difficult without routine analytical testing. Next, another study has emerged showing that micro and nanoplastics may strengthen biofilms, potentially making pathogenic bacteria more difficult to eradicate. This time, the research focused on biofilms in drinking water infrastructure. The study examined dual-species biofilms comprising E. coli and Pseudomonas aeruginosa exposed to positively and negatively charged polystyrene nanoplastics at environmentally relevant concentrations. The researchers found that nanoplastics entered bacterial cells and increased oxidative stress, raising reactive oxygen species levels by approximately 2.2-fold. This stress activated dormant bacteriophages, triggering partial lysis of E. coli cells and releasing intracellular material into the surrounding biofilm. At the same time, the nanoplastics activated bacterial stress responses and enhanced quorum sensing, stimulating bacteria to produce larger amounts of extracellular polymeric substances, or EPS, which comprise a biofilm's protective matrix. Together, these processes increased extracellular DNA within the biofilm and improved its mechanical strength by approximately 1.5-fold. Importantly for drinking water safety, these strengthened biofilms also showed significantly increased resistance to chlorine disinfection. The authors concluded that stronger, more disinfectant-resistant biofilms could create new challenges for drinking water treatment and distribution systems by making microbial communities more difficult to eradicate from infrastructure surfaces. The findings add to emerging evidence suggesting that micro and nanoplastics can influence bacterial behavior in ways that affect microbial persistence, antimicrobial resistance, and virulence. Turning to food processing environments, a new study examined how plastic food contact materials, surface design, and background microorganisms influence the persistence of Listeria monocytogenes biofilms. Researchers from the University of Florida, USDA's Agricultural Research Service, and collaborating institutions evaluated 4 plastics commonly used in food processing applications: ultra-high molecular weight polyethylene, polyoxymethylene, polypropylene, and polyvinyl chloride, or PVC. Each material was tested with smooth, microdot, and microline surface topographies under both single-species and multi-species biofilm conditions. The researchers found that plastic composition and the presence of other microorganisms had the greatest influence on L. monocytogenes persistence. Among the materials tested, polypropylene consistently supported greater L. monocytogenes accumulation in multi-species biofilms, while line-pattern surfaces generally promoted higher pathogen persistence than smooth surfaces under mixed species conditions. Background microorganisms also had important effects. Pseudomonas fluorescens consistently promoted L. monocytogenes biofilm formation across nearly all plastic types and topographies. Meanwhile, E. coli 0157 and Ralstonia insidiosa produced either synergistic or antagonistic effects depending on the specific surface. Notably, conventional indicators such as surface roughness and wettability did not reliably predict L. monocytogenes biofilm formation. The authors said their findings demonstrate that pathogen persistence on plastic food contact surfaces is governed by the combined effects of material composition, surface topography, and microbial ecology rather than any single factor. Finally, A study has highlighted the combination of stress-adaptation traits that may enable environmental E. coli to persist throughout the farm-to-fork continuum. The study analyzed 18 E. coli isolates collected from irrigation water, soil, and lettuce grown in the Litani River Basin region of Lebanon. Researchers evaluated isolates for antibiotic susceptibility, biofilm formation, and tolerance to acetic acid, and conducted whole genome sequencing to identify virulence-associated genes, antimicrobial resistance determinants, and plasmids. Although most isolates remained susceptible to the antibiotics tested, 11% were classified as multi-drug resistant. In contrast, moderate to strong biofilm formation was observed in 83% of isolates, and several demonstrated reduced susceptibility to acetic acid concentrations comparable to those used in household produce washing. The researchers also identified significant positive correlations among acid tolerance, biofilm formation, and antibiotic resistance, suggesting that these stress adaptation traits frequently occurred together. However, neither biofilm formation nor acid tolerance could be linked to individual genetic determinants, indicating that the traits were likely influenced by multiple regulatory and environmental factors. The authors emphasized that the isolates generally did not exhibit high-risk clinical resistance profiles. Still, the researchers argued that the co-occurrence of biofilm formation, acid tolerance, and antimicrobial resistance genes could enhance bacterial survival and complicate microbial control efforts in produce production systems. The researchers concluded that the findings support integrated, preventive food safety strategies that reduce contamination in irrigation water, soil, and other environmental reservoirs before produce reaches consumers. For more information on any of the stories mentioned in this video, be sure to check out the Food Safety Magazine coverage linked in the description. Check back on the third Tuesday of each month for the latest Food Safety 5 episodes, available at food-safety.com or on our YouTube channel.